Hello everyone, I'm Xiaohong Liang from Department of Immunology, Shandong University. In this lecture, we are going to talk about the development of tumor immunology. Cancer, everyone knows this about the word, right? It brings such a painful experience and heavy financial burden for the patient and the family that people have a terrible feeling even hearing about this word. Do you? I do. This slide shows the trend in new cancer cases and deaths from 2000 and 2011. We can see that both for men and women, the cancer occurrence and death rate is continuously and stably increasing year by year. Worldwide, cancer is the second leading cause of death. 4 people die from cancer every second. The situation seems to be worsening in the near future, and human beings will be continuously threatened by cancer. Thus, how to find out an efficient way to treat and eliminate cancer is a big issue, either for basic research or for clinical practice. Why? Because based on the clinical dogma for tumor, transformation into tumor cells owes to the intrinsic alteration of normal cells induced by carcinogen or inherited genetic mutations. However, recent advancements in tumor indicate that this model seems to oversimplify the progression of tumor. In 2000, Professor Weinberg, a very famous cancer expert, summarized tumor cell biology with 6 characteristics as shown in this chart. However, with the immunofluorescence staining, researchers found that in human colon cancer tissues, there are not only tumor cells, but also a huge number of stroma cells, endothelial cells, extracellular matrix protein, as well as a variety of immune cells. Now we know that tumor cells do not equal to tumor. Tumor cells, together with many kinds of other components, set up a complicated microenvironment to support tumor progression. Thus, research on tumor should not be only limited to tumor cells themselves. Actually, it is also the main reason for the great difficulties to study the pathogenesis of cancer and to cure the cancer. More importantly, clinical analysis disclosed that different tumors showed distinct immune cell atlas. As shown in this picture, in non-metastatic tumors, the activated and effector T cells are the major infiltrated immune cells. However, the inhibitory immune subsets like Treg, M2 macrophages would be more rich in metastatic tumor tissues. 10 years later, in 2011, it was updated and tumor cells are endowed with 4 other hallmarks. From then on, immune cells or inflammation in tumor development is more and more emphasized, and tumor immunology becomes a hot field. Accordingly, immunotherapy becomes one of important strategies for eradicating tumor. In 2013, the approval of blocking antibody for CTLA-4 and PD-1 by FDA and CAR-T or CAR-NK, a promising strategy for T-cell, NK cell reforming, promotes immunotherapy to be a great breakthrough for tumor biotherapy. This is a landmark event in cancer immunology. In general, tumor immunology is mainly to study the immunogenicity of tumor and the mechanism of immune response to tumor. To demonstrate the relationship between the status of immune system and the generation development of tumor, to explore the method for tumor diagnosis, therapy, and prevention. Timeline of the history of tumor immunology from its inception in the late 19th with William Coley's experiments to modern-day tumor immunology with the FDA approval of 2 immunotherapies for treatment of cancer. Based on current clinical outcomes and laboratory-based research using animal models, future promising therapies will be combining checkpoint blockade, co-stimulation alone, or in combination with conventional approved treatments. William Bradley Coley, an American bone surgeon and cancer researcher, found that erysipelas infection helped sarcoma disappearing in clinics. Then he tried to inject Streptococcus bacteria to the patients. Then, for a long time, scientists tried to establish an animal model to explain the mechanisms about Coley's toxin, but the progress was not so optimistic. Until 1970s, Burnett and Medwa established a mouse model and made a breakthrough. They proposed the immune surveillance theory, which is all we know, very important immune concept. They established a series of mouse tumor models. The mouse was firstly induced to grow tumor by chemical carcinogen. Then the tumor tissues were cut, and the tumor cells were transplanted into the original tumor-bearing mice or to another syngeneic mice. You can see that the original mice is resistant to these tumor cells. Well, they grow well in syngeneic mice. But if the tumor cells were transplanted together with the CD8 positive T cells from original tumor-bearing mice, the tumor cells were killed. This was also called immune surveillance against tumor. Over the past 2 decades, numerous studies have established that the immunodeficient mice are more tumor-prone than are immunocompetent mice after treatment with carcinogens such as MCA. The immunodeficient mice tested in such experiments include gene-targeted mice on pure genetic backgrounds with defects of innate or adaptive immunity. Then another experiment was performed. Tumor cell lines were established from tumor arising in competent mice and immunodeficient mice, and these cells were then injected into immunocompetent wild-type recipient mice. It was found that 100% of tumors from carcinogen-treated wild-type mice formed progressively growing tumors in naive genetic immunocompetent mice. In contrast, although only half of tumor cells from carcinogen-treated immunodeficient mice were capable of forming progressively growing tumors in naive syngeneic immunocompetent recipients in purple, whereas the other half of the cell lines were rejected by the recipients in red. Thus, tumors from immunodeficient mice are termed unedited phenotype. The above experiment further develops the cancer immunoediting concept, which describes 3 sequential phases for tumor development, including elimination, equilibrium, and escape. In the elimination phase, innate and adaptive immunity work together to destroy developing tumors long before they become clinically apparent. Many of the immune molecules and cells that participate in the elimination phase have been identified. If this phase goes to completion, then the host remains free of cancer, and elimination thus represents the full extent of the process. If, however, a rare cancer cell variant is not destroyed in the elimination phase, it may then enter the equilibrium phase, in which its outgrowth is prevented by immunologic mechanisms. T cells, IL-12, and interferon gamma are required to maintain tumor cells in a state of functional dormancy. Editing of tumor immunogenicity occurs in the equilibrium phase. Equilibrium may also represent an end stage of the cancer immunoediting process and may restrain outgrowth of occult cancers for the lifetime of the host. However, as a consequence of constant immune selection pressure placed on genetically unstable tumor cells held in equilibrium, tumor cell variants may emerge that are no longer recognized by adaptive immunity, for example, antigen loss variants, or tumor cells that develop defects in antigen processing or presentation, become insensitive to immune effector mechanisms, or induce an immunosuppressive state within the tumor microenvironment. These tumor cells may then enter the escape phase, in which their outgrowth is no longer blocked by immunity. These tumor cells emerge to cause clinically apparent disease. In summary, today we know that what is tumor immunology and the cancer immunoediting theory.